Welcome, everyone, and thank you for being here today. I'm excited to introduce you to our presentation titled Wastewater Biology, the Microlife. This is a comprehensive guide to understanding the fascinating microscopic organisms that play a crucial role in biological wastewater treatment and water quality management. Over the course of this presentation, we'll explore who these microbes are, what they do, and why they are essential for maintaining clean water. So, let's dive in and uncover the hidden world of wastewater biology together. Let's talk about the invisible workforce that keeps our water clean. What are microorganisms? Well, these are tiny life forms that are so small you can't see them with the naked eye. They range in size from about 0.2 micrometers up to several millimeters, which is still incredibly small. These microscopic organisms are actually the unsung heroes of wastewater treatment. They perform the crucial job of breaking down organic matter and recycling nutrients, helping to purify water before it's returned to the environment. When we say microorganisms, we're referring to a diverse group called Microlife, which includes bacteria, protozoa, and even metazoa like rotifers and nematodes. Each of these groups plays specialized roles within the complex ecosystem of a water resource recovery facility. It's like a tiny, intricate community working behind the scenes to keep our water safe. Now, let's step back in time a bit. The microscopic world remained hidden until the 1660s, when Robert Hooke built the first crude microscope. That was a huge breakthrough because it allowed us to start seeing these tiny organisms for the first time. Then, Anton van Leeuwenhoek took it a step further. He was the first to study and describe bacteria and protozoa in detail, calling them animalcules. This discovery was revolutionary. It changed how we understood many things: disease, food spoilage, fermentation, and laid the foundation for microbiology. By the 1880s, scientists had identified bacteria as the cause of cholera and realized that epidemics like typhoid were linked to water contaminated with microbiological agents. So, understanding these microorganisms was key to improving public health and sanitation, and it still underpins how we treat wastewater today. Let's explore the four primary groups of microorganisms that play a vital role in wastewater treatment. First, we have bacteria. These are single-celled organisms found in huge numbers, especially in polluted water. They come in various sizes and shapes and are the most prolific microorganisms on Earth. In wastewater treatment, bacteria perform the majority of the work. They break down organic matter. Some bacteria form long, thread-like filaments, while others create clusters called floc, which are essential for settling solids. Next, we have protozoa. These are larger single-celled organisms, usually much bigger than bacteria. Many are mobile and swim rapidly through their microscopic environment, while others attach themselves to particles. Protozoa are crucial because they help clarify the effluent by preying on bacteria, which helps control bacterial populations. They also serve as useful indicators for evaluating the health of the treatment process. Moving on to metazoa, these are multi-celled animals, including rotifers. Rotifers are fascinating. They have rotating crowns of tiny hair-like structures called cilia. Although larger than bacteria and protozoa, they're still microscopic. They play an active role in breaking down organic waste in wastewater. Other metazoa include nematodes, water bears, and bristleworms, all contributing to the treatment process in their own ways. Finally, we have viruses. These are much smaller than bacteria and are invisible under regular microscopes. Viruses are parasites. They need to infect host cells to reproduce. While they don't have a direct role in wastewater treatment, it's important to remember that many diseases caused by viruses, like hepatitis, can be transmitted through polluted waters. Recognizing these microorganisms helps us understand the complexity of wastewater treatment and the importance of controlling pathogens. Let's explore the microscope, which truly is a window to the invisible world. Microscopes are crucial tools in wastewater microbiology because they help us identify and count microorganisms that are otherwise invisible to the naked eye. There are two main types of microscopes we use, each serving different purposes in our work. First, we have the compound microscope. This type offers high magnification, typically from 40x up to 1000x, thanks to its complex lens system. It has objective lenses with magnifications of 4x, 10x, 40x, and 100x, and usually a 10x ocular lens. When you combine the ocular and objective lenses, you get the total magnification. For example, a 10x ocular with a 40x objective gives you 400x total magnification. It's important to note that the 100x objective often requires immersion oil to achieve the best resolution, which is about 200 nanometers when used correctly. Next, we have the stereoscopic binocular microscope. This one provides lower magnification, ranging from 8x to 40x, but it offers a true three-dimensional view through two complete lens systems. It's especially useful for examining larger specimens that are too thick for the compound microscope, or for objects that are too small to see clearly with the naked eye. It uses reflected light instead of transmitted light, and features like variable zoom and a long working distance make it ideal for manipulating and observing specimens simultaneously. Both microscopes are invaluable in wastewater biology. The compound microscope allows us to see tiny microorganisms in great detail, while the stereoscopic microscope helps us work with larger or more complex samples. Together, they give us a comprehensive view of the microscopic world that influences wastewater treatment processes. Bacteria are often considered the primary workers in the treatment process because of their crucial role in recycling nutrients and breaking down waste. They are prokaryotic, single-celled organisms that belong to the kingdom Monera, and their sizes typically range from about 0.2 to 2 micrometers. Interestingly, they make up roughly 90 to 95% of the biomass in activated sludge, which highlights just how vital they are in ecosystems and treatment systems alike. When it comes to their shapes, bacteria mainly fall into three basic categories: cocci, which are spherical, bacilli, which are rod-shaped, and spirilla, which have a spiral form. But there are also some unusual types worth noting, like sheathed bacteria such as Spheratillus, stalked bacteria like Colobacter, budding bacteria like Hyphomicrobium, and filamentous actinomycetes such as Nocardia. These variations often have specific roles or adaptations that help them thrive in different environments. Now, moving on to their energy and oxygen needs. This is fascinating because it influences how bacteria function in treatment systems. Some bacteria are autotrophs, meaning they use carbon dioxide as their energy source, while others are heterotrophs and rely on organic compounds. Regarding oxygen, some bacteria are aerobes, requiring oxygen to survive. Others are facultative anaerobes, which can adapt to both oxygen-rich and oxygen-poor conditions, and some are strict anaerobes that can't tolerate oxygen at all. Temperature is another important factor. Bacteria have different temperature preferences. Psychrophilic bacteria thrive in cold environments from 0 to 20 degrees Celsius. Mesophilic bacteria prefer moderate temperatures, around 25 to 40 degrees, and thermophilic bacteria are found in hotter conditions, above 45 degrees Celsius. These adaptations help bacteria to operate efficiently in various settings. Let's also touch on their cell structure. The cell wall gives bacteria their shape and structural integrity. Inside, the cytoplasmic membrane controls what enters and exits the cell. The glycocalyx, or capsule, helps bacteria attach to surfaces and offers some protection. Flagella are like little propellers that enable movement, which is vital for bacteria to seek nutrients or disperse. They store energy in forms like sulfur granules or polyhydroxybutyrate, or PHB. And some bacteria form endospores. These are tough, dormant structures that resist harsh environmental conditions, ensuring their survival through tough times. Understanding these characteristics helps us appreciate how bacteria work as the primary workers in wastewater treatment, and why their diversity and adaptability are so important in maintaining system efficiency. Let's take a closer look at the origin and dominance of the bacteria involved in wastewater treatment. These bacteria naturally occur in aquatic systems and soils, which means they are part of the natural environment. Originally, wastewater treatment relied on these natural processes, where sewage discharged into surface water bodies was naturally filtered and treated by the existing microbial community. However, as industrialization and urban populations grew after the Civil War, these natural systems became overwhelmed and couldn't keep up with the increasing waste loads. This challenge led to the development of mechanical treatment methods to supplement or replace natural processes. Starting around 1900 to 1920, mechanical treatment began with basic steps like screening and settling, known as primary treatment. During this period, fixed media biological systems emerged, seeded with sludge from streams or rivers to introduce beneficial bacteria. Between 1920 and 1930, biological secondary treatment methods, especially fixed media types, became widely used in the United States. During this time, studies on the assimilative capacity of streams were conducted, as many still received raw wastewater directly. From 1920 onward, activated sludge systems were developed to handle larger flows. These systems were seeded from earlier fixed media processes or existing activated sludge facilities. Interestingly, today's systems can even operate without initial seeding, although this makes the process slower at first. It's important to understand that the environment in which these bacteria grow plays a crucial role in determining which species dominate. Conditions that favor certain bacteria will lead to their proliferation. As conditions change, so does the microbial community, a process called succession. In secondary treatment, we create highly oxygenated environments that favor aerobic and facultative anaerobic bacteria. These microorganisms are essential because they metabolize organic matter, converting it into bacterial biomass. This biological activity is key to effectively cleaning wastewater and highlights the importance of understanding bacterial ecology in our treatment systems. Protozoa are fascinating microscopic organisms that are key players in biological treatment processes. They are eukaryotic, mostly single-celled, and heterotrophic, meaning they consume organic material rather than producing it themselves. Their size varies quite a bit, from about 5 to 1,000 micrometers. Interestingly, in activated sludge, you can find around 50,000 protozoa per milliliter, which makes up roughly 5% of the dry weight of suspended solids. This highlights how vital they are in wastewater treatment and also makes them useful as indicators of effluent quality. Now, within protozoa, we have different types, each with unique features. Let's start with flagellates. These organisms have one or more flagella, those long, hair-like structures that help them move and feed. Some flagellates contain chlorophyll, making them photoautotrophic, and they play significant roles in oxidation lagoons by contributing to the microbial community. Others are colorless and heterotrophic, obtaining food either as particles or dissolved substances. Next, we have amoebae. They move and feed using pseudopodia, these retractable arm-like extensions. Amoebae can be naked, meaning without a shell or testate, with shells made of proteins, silica, calcium carbonate, or other materials. These shells can vary, but both naked and shelled amoebae are fairly common in wastewater treatment processes. Finally, let's look at ciliates. They are distinguished by having cilia, tiny hair-like structures, covering their bodies in organized rows called chyintes. Ciliates also have two types of nuclei and undergo a unique form of sexual reproduction called conjugation. They are considered the most important protozoa in treatment because of their role in controlling bacteria and maintaining the microbial balance. Examples include groups like gymnostomes, hymenestomes, hypotrichs, and paratrichs, all of which contribute significantly to the treatment ecosystem. So, understanding these protozoa and their roles helps us appreciate the complexity and efficiency of biological wastewater treatment systems. Let's take a closer look at how protozoa feed and reproduce. There are three main feeding strategies to consider. First, we have photoautotrophy. This is where flagellates absorb sunlight and fix carbon dioxide, essentially acting as primary producers. They need light, inorganic ions, and trace elements, which are all present in wastewater. Keep in mind, though, that light is often the most limiting factor for these organisms. Next is osmotrophy. Many flagellates utilize organic complexes dissolved in the wastewater. They may also require vitamins produced by bacteria, which they often compete for with heterotrophic bacteria. Then there's phagotrophy, where protozoa ingest particulate food matter. Most of these are bacterivores, feeding on bacteria that are either dispersed or attached to surfaces. Some protozoa are saprotrophs, feeding on detrital matter, while others are carnivores, preying on other protozoa. It's important to note that not all bacteria make a complete diet when supplied alone, which influences their feeding behavior. Now, turning to growth and reproduction. The typical life cycle involves growth followed by binary fission, which is a form of asexual reproduction. In this process, the cell divides into two new individuals, passing on the biomass to the next generation. There are other forms too, like budding, which involves unequal fission and multiple fission, where many daughter cells are produced at once. Most protozoa also undergo sexual reproduction, adding to their reproductive strategies. Finally, when protozoa feed on bacteria, their growth and reproductive rates tend to increase as food availability rises. For example, in activated sludge, ciliates can divide every 4 to 5 hours, showing rapid reproduction. In contrast, testate amoebae grow much more slowly, often measured over days. Understanding these feeding and reproductive strategies helps us appreciate how protozoa thrive in wastewater environments and contribute to treatment processes. Let's take a closer look at the spatial distribution and succession of organisms in these wastewater treatment processes. In fixed growth systems like trickling filters and rotating biological contacts, or RBCs, you'll notice that different types of microorganisms dominate at various points along the system as the water gets purified. Starting at the surface or inlet, you'll find a community mainly made up of flagellates and amoebae. These organisms are usually near the surface, where soluble organic substrates are more accessible, and the environment tends to have lower dissolved oxygen levels. Moving to the middle section, the diversity increases. Here, ciliates become more common, and bacterivores, those that feed on bacteria, are predominant. Conditions are transitional, with more oxygen than at the inlet but still some organic material present. At the bottom or outlet, the community shifts again. Now, carnivores take over, feeding on other protozoa. This zone has higher dissolved oxygen levels and less organic material, resulting in the most diverse protozoan community of the entire system. Switching gears to activated sludge systems, there's a clear temporal succession during maturation. Initially, you see colorless flagellates dominating. As time progresses, their numbers decrease and free-swimming ciliates become more prevalent, peaking around 20 days. Following that peak, crawling ciliates like Aspidisca and euplotes take over. Finally, attached paratrichous ciliates such as Vorticella, Epistoles, and Opercularia become dominant. This succession pattern reflects a combination of nutritional preferences and changing environmental conditions, which is crucial for maintaining effective treatment. Understanding these dynamics helps us optimize the process and ensure the health of the microbial community involved in wastewater purification. Factors affecting protozoan populations. Let's start with dissolved oxygen. This is one of the most critical factors that determines whether certain protozoan species are present or absent in a given environment. We can think of species as falling into 3 main groups: those that require free oxygen, those that can use it when it's available, and those that actually cannot grow in its presence. Typically, aerobic processes are maintained at around 1 to 2 milligrams per liter of oxygen. However, in some stages like sedimentation tank bottoms, anaerobic conditions can prevail, influencing which protozoa can survive there. Next, let's talk about pH and carbon dioxide. These are closely linked because the concentration of carbon dioxide and the local geochemistry influence each other. You'll often find chemical gradients around sludge or biofilm, which can impact protozoan populations significantly. For example, in oxidation lagoons, higher pH levels might explain why we see fewer protozoa compared to algae. And since carbon dioxide is a primary carbon source for photosynthetic flagellates, High concentrations can become toxic to many protozoa. Moving on to light, this is the energy source for photosynthetic organisms, but its penetration is quite limited in some treatment processes. In trickling filters, light only penetrates a few millimeters, and in activated sludge mixed liquor, suspended solids block light altogether. This limited light explains why we find very few photosynthetic protozoa in activated sludge, and why those that do exist are usually confined to the upper layers of biological filters. Now, on to flow rate. This affects how quickly the liquid moves through the treatment system, impacting the dilution rate and the velocity of water over surfaces. Higher flow rates lead to greater washout of free-swimming protozoa, reducing their populations. For protozoa attached to sludge flocks, the mean cell residence time is equally important. If water moves too quickly, they might be sloughed off or washed away, affecting overall community structure. Let's discuss toxic waste. Industrial wastewaters, often combined with domestic waste, can introduce heavy metals and other toxic substances that impact protozoan communities. The toxicity depends on factors like pH, dissolved oxygen, mean cell residence time, temperature, and the presence of other toxic compounds. It's important to note that the actual concentrations that cause serious inhibition are difficult to state precisely, as it varies case by case. Finally, predation plays a significant role. Many processes contain predatory ciliates, like those preying on sessile paratrichs, and suctoria that feed on a wide range of protozoa. It's not just ciliates. Many hypotrichs ingest flagellates, and amoebae feed on both ciliates and flagellates as well. Interestingly, there's still a lot we don't fully understand about the kinetics of protozoan predation, but it clearly influences community dynamics within treatment systems. All of these factors, oxygen, pH, light, flow, toxins, and predation, interact in complex ways to shape protozoan populations, which are vital indicators of treatment performance and system health. Let's take a closer look at the role of protozoa in treatment processes. Protozoa influence effluent quality in several key ways. The most important are their ability to remove bacteria through predation and to enhance floc formation, which helps clarify the water. They also stimulate bacterial growth, support healthy floc formation, and assist in breaking down organic waste. One striking point is their impact on E. coli removal. When ciliates are present, about 95% of E. coli are eliminated, compared to only 50% when protozoa are absent. This shows how effective protozoa are in reducing pathogenic bacteria naturally. In terms of bacterial consumption, a single ciliate like Tetrahymna piriformis can consume up to 500 bacteria per hour. That's a significant contribution, and it explains how they help keep bacterial populations in check. Protozoa also make up roughly 5% of the dry weight of suspended solids in activated sludge. Though it might seem small, their role is quite impactful in maintaining overall process efficiency. Experiments with pilot-scale activated sludge systems clearly show the difference protozoa make. Without ciliates, the effluent tends to be highly turbid and of lower quality, mainly because of the large number of suspended bacteria. When protozoa are present, the effluent clarity improves dramatically, with a notable decrease in viable bacteria. It's also important to note that protozoa feed on a wide range of bacteria, including fecal coliforms and pathogenic bacteria responsible for diseases like diphtheria, cholera, typhus, and streptococcal infections. Their continual grazing stimulates the growth of healthy bacteria, which helps form active flocks. This process accelerates purification and makes it more thorough, especially when ciliates are involved. So overall, protozoa are essential allies in biological treatment. They naturally help improve water quality, speed up the removal of harmful bacteria, and support the stability of the treatment process. Rotifers are fascinating wheel-bearing animals that live in aquatic environments. There are about 2,000 known species, and their name comes from Latin words rota meaning wheel and fere meaning to bear, highlighting their distinctive wheel-like structures. These structures are called corona, which are disc-shaped and covered with tiny cilia that constantly move. This movement helps them gather food particles and also assist with locomotion. Size-wise, rotifers range from just 40 micrometers up to 500 micrometers, and their typical lifespan is between 15 and 45 days. Most of them are colorless, except for their eye spot, although sometimes the food they ingest can give them a slight color. Interestingly, they can reproduce in several ways: sexually, asexually, or through an alternating process, which adds to their adaptability. Now let's look at their body structure. They have three main regions: the head, the trunk, and the foot. The head includes the corona, the mouth opening, and a mastax, which contains jaws called trophus. The trophus is a hard structure inside the buccal cavity that helps filter and grab food. The trunk is where most of the internal organs are housed, like the esophagus, stomach, intestines, gonads, and the anus. The foot is a muscular part, sometimes equipped with toes that help them attach to surfaces, although some rotifers lack this feature entirely. There are two primary classes of rotifers. The first is monogononta, which typically has one gonad and is covered by a protective shell called lorica. Male rotifers are present but usually short-lived and rarely seen. The second class is digononta, also known as Bedeludia, which has two gonads, a well-developed corona, and a cylindrical body capable of contracting. These rotifers can survive in low-oxygen environments and reproduce solely through asexual means, with no males present. Understanding these differences helps us appreciate how diverse and adaptable rotifers are within their aquatic ecosystems, playing a vital role in their environments and exemplifying the complexity of microscopic life. Rotifers play a crucial role in treatment processes by helping to stabilize organic wastes through a variety of beneficial actions. They stimulate microbial activity, improve oxygen penetration, and assist in recycling mineral nutrients. Even though they are rarely found in large numbers, usually only 1 to 4 species in well-operated facilities, they have hearty appetites that make up for their limited numbers. In oxidation lagoons, rotifers mainly feed on phytoplankton or algae. By continuously cropping algae, they help keep the algal populations healthy and also recycle essential mineral nutrients. For example, each Brachynes calcareus can consume over 12,000 phytoplankton cells daily, which significantly contributes to high-level wastewater treatment. In fixed growth processes, their primary role is to control biological slime layers by snipping off the top layer. This activity allows oxygen to penetrate deeper into the layers, increasing aeration and promoting microbial stabilization of organic waste. Additionally, keeping slime layers porous reduces ponding issues in trickling filters. When it comes to activated sludge systems, rotifers help maintain the health of the bacterial community by consuming large amounts of bacteria. They also reduce secondary effluent turbidity by eating non-flock-forming bacteria and promote better flock formation by snipping off particles to expose new surfaces for bacteria to adhere to. Their mucus secretion aids in this flock formation, acting as a bonding site for bacteria and waste material. Finally, rotifer populations serve as valuable process control indicators. Their types and abundance are influenced by factors like dissolved oxygen, mean cell residence time, suspended solids, pH, temperature, and BOD levels. Because rotifers are in ecological balance with their environment, changes in environmental conditions directly affect their populations. Monitoring their numbers, types, and activity provides an early warning system for potential process problems, such as the entry of toxic substances. Let's take a closer look at some of the larger, more complex animals we encounter in wastewater treatment, which fall under the category of Metazoa. As you might remember, Metazoa refers to all multicelled animals, but in our context, we're focusing on those that are bigger and more developed than Protozoa. These include nematodes, water bears, bristleworms, water fleas, and seed shrimp. Starting with nematodes, or roundworms, which belong to the phylum Nematoda. These are often microscopic and commonly found in wastewater treatment systems. They have elongated, cylindrical bodies that taper at both ends, and they can range from about 0.5 millimeter up to 3 millimeters long. Free-living nematodes feed on bacteria, algae, protozoa, and organic debris, making them important players in the ecosystem. They typically reproduce sexually, with separate sexes, and their life cycle includes an egg stage, 4 juvenile stages, and then the adult. Nematodes are quite beneficial because they help control bacterial populations and assist in breaking down organic matter. Next, we have water bears, or tardigrades, which are even more fascinating. These tiny creatures, measuring between 50 and 1,200 micrometers, have 4 pairs of stubby legs ending in claws, and they move with a slow, bear-like gait. They feed on plant cells, bacteria, and other small organisms. What makes tardigrades particularly remarkable is their ability to survive extreme conditions through a process called cryptobiosis. They can withstand desiccation, freezing, high temperatures, and radiation, conditions that would be lethal to many other forms of life. You'll often find them in activated sludge and trickling filters, playing a vital role in those environments. Finally, let's discuss bristleworms, or oligochaetes, which are segmented worms belonging to the phylum Annelida. These worms have bristles, or setae, on each segment, which help them move and anchor themselves. In wastewater treatment, aquatic oligochaetes like Eolysoma are common. They feed on bacteria, algae, and organic debris, contributing significantly to breaking down organic matter. They can reproduce either sexually or asexually through budding, and their activity helps maintain healthy bacterial populations, which is essential for the overall efficiency of the treatment process. All of these animals, although small, play a crucial role in maintaining the balance and health of wastewater treatment ecosystems. They help control bacterial populations, decompose organic material, and contribute to the stability of the system. Now, let's take a closer look at some additional metazoan groups that play important roles in wastewater treatment. First, we have the gastrotrichs. These are tiny, microscopic animals, ranging from about 50 to 800 micrometers in length, with elongated bodies covered in scales or spines. They move around using cilia on their ventral surface, which helps them glide through their environment. Gastrotrichs feed primarily on bacteria, algae, and organic particles, making them key players in controlling bacterial populations. You'll often find them in activated sludge and trickling filters, where they help maintain a balanced microbial community. Next, we have water fleas, scientifically known as Cladocera. These are small crustaceans, typically between 0.2 and 3 millimeters long, with a laterally compressed body enclosed in a transparent carapace. They swim using their large second antennae. Their diet consists of bacteria, algae, and organic matter, and interestingly, they can reproduce asexually through parthenogenesis when conditions are favorable. Water fleas are commonly found in oxidation lagoons and stabilization ponds, where they contribute to organic matter breakdown. Moving on, seed shrimp or ostracods are another group of tiny crustaceans, measuring about 0.5 to 2 millimeters. Their bodies are completely enclosed in a bivalved carapace that resembles a tiny clam. They swim or crawl using their appendages and feed on bacteria, algae, and detritus. Like water fleas, they are also found in oxidation lagoons and stabilization ponds, playing a role in organic matter decomposition. Finally, we have water mites, or hydrocarina. These small arachnids are about 0.5 to 5 millimeters long, with a globular body and 4 pairs of legs. They swim by moving their legs and are mostly predators, feeding on small invertebrates. Water mites are typically found in trickling filters and oxidation lagoons, and their presence indicates well-oxygenated conditions and a mature treatment process. Overall, these metazoan groups contribute significantly to wastewater treatment by preying on bacteria, breaking down organic matter, and supporting healthy microbial communities. Their presence and abundance are valuable indicators of the overall health and efficiency of the treatment process. Filamentous organisms are bacteria that grow as long, thread-like structures extending from activated sludge flocks. Now, while some filamentous growth actually plays a beneficial role in helping form these flocks, It's important to understand that too much growth can cause serious operational issues. Excessive filamentous growth can lead to bulking and foaming, which disrupts the treatment process. On the beneficial side, these filamentous bacteria provide a sort of structural backbone for flock formation. They help enhance the strength and settleability of the flocks, making it easier for solids to settle out effectively. They also increase the surface area for bacterial attachment, which improves the overall treatment efficiency. Plus, they facilitate oxygen transfer into the interior of the flock and help transport nutrients within the flock structure, supporting the biological activity necessary for wastewater treatment. However, problems arise when filamentous growth becomes excessive. Overgrowth interferes with flock settling, causing bulking, which can reduce the capacity of clarifiers. It can also lead to foam and scum formation on the surface, which complicates operation and affects the quality of the effluent. When this happens, solids can be carried over into the treated water, decreasing overall effluent quality. Now, there are several common types of filamentous bacteria you might encounter. For example, Spheratillus natans forms sheathed filaments and tends to thrive under low dissolved oxygen or nutrient-deficient conditions. Type 21N has branched filaments and is often associated with low oxygen levels and septic conditions. Thiothrix stores sulfur and appears in sulfide-rich wastewaters. Microthrix parvicella has coiled filaments and is typical in cold temperatures with high fat content. Lastly, Nocardia forms branched filaments that can cause foaming issues, especially when the process involves long cell residence times. Understanding these different types and their behaviors is key to managing filamentous organisms effectively in the treatment process. It's all about striking the right balance, leveraging their beneficial roles, while controlling their overgrowth to prevent operational problems. Controlling filamentous bulking is essential for maintaining effective wastewater treatment. When filamentous organisms extend from flocks, they interfere with settling, leading to poor sludge settling performance. You'll notice this when the sludge volume index, or SVI, rises above 150 milliliters per gram, which indicates that settling is becoming inefficient. Our control strategies focus on reducing the factors that encourage filamentous growth. First, operational adjustments are key. We can manipulate the return-activated sludge rate to maintain a proper solids inventory, which helps keep the microbial community balanced. Reducing the solids loading on the clarifier can also improve settling. Increasing dissolved oxygen levels in the biological reactor favors bacteria that form good flocks, rather than filamentous organisms. And adjusting the mean cell residence time ensures we optimize bacterial populations for better settling. Next, chemical addition can be effective. Using flocculating agents like polymers or metal salts helps improve the aggregation of solids. In cases where filamentous organisms are problematic, applying toxic chemicals such as chlorine, hydrogen peroxide, or ozone can selectively target and kill these organisms. Chlorine is the most common choice, but it must be carefully dosed to avoid harming beneficial bacteria. Hydrogen peroxide and ozone offer alternative mechanisms that can be more selective. Regarding wastewater composition, adding nutrients like nitrogen and phosphorus can be necessary if they're deficient, as these support healthy bacterial growth. Removing sulfides, especially when they're in high concentrations, helps reduce filamentous growth. Also, adjusting the pH to an optimal range, between 6.5 and 8.5, promotes the activity of flock-forming bacteria. Ensuring a balanced ratio of nutrients encourages the desired bacteria to thrive over filamentous organisms. Finally, process modifications can make a big difference. Changing the influent feeding points or patterns can create conditions that favor good flock formation. Installing selectors, whether aerobic, anoxic, or anaerobic zones, at the process head can give flock formers a competitive advantage. These selectors influence substrate uptake kinetics, making it easier for beneficial bacteria to dominate and suppress filamentous growth. All these strategies are interconnected and aim to create a balanced environment that promotes good settling and overall process stability. Let's talk about indicator bacteria and pathogens. Indicator bacteria are used as stand-ins for pathogenic organisms when we're monitoring water quality. Since testing for every possible pathogen is impractical, we rely on these indicator organisms that correlate with fecal contamination to give us a good idea of potential health risks. First, we have total coliforms, which include bacteria like Escherichia coli and related species. These bacteria are found in the intestines of warm-blooded animals as well as in the environment. Their presence suggests there might be fecal contamination somewhere in the water. Next, we look at fecal coliforms, which are a subset of total coliforms. They come specifically from the intestinal tracts of warm-blooded animals and are a more accurate indicator of fecal contamination. Interestingly, about 90% of fecal coliforms are E. coli. We test for these at higher temperatures, around 44.5 degrees Celsius, to distinguish them from environmental coliforms that might be naturally present. Then there's fecal streptococci, which are bacteria from the intestinal tract that tend to be more resistant to environmental stresses than coliforms. Because of their resilience, they're useful for assessing older or more persistent contamination. The ratio of fecal coliforms to fecal streptococci can also help us determine whether the contamination source is human or animal. Now, within fecal streptococci, we have enterococci. These are a specific subgroup and serve as a more precise indicator of fecal contamination. Interestingly, they tend to persist longer in the environment than coliforms and have a stronger correlation with illness related to recreational water use. That's why the EPA recommends using enterococci for recreational water quality standards. Lastly, let's touch on pathogenic organisms. Wastewater can contain a variety of harmful pathogens, bacteria like Salmonella, Shigella, Vibrio, viruses such as hepatitis A, rotavirus, norovirus, protozoa like Giardia and Cryptosporidium, and even parasitic worms. While treatment processes significantly reduce these pathogens, they may not eliminate all of them completely. Disinfection adds an important barrier, but it's crucial for operators to wear protective equipment and follow safety procedures when handling wastewater to prevent infection. Overall, understanding these indicator bacteria and the pathogens they help us detect is vital for protecting public health and ensuring water safety. Now, let's discuss microscopic examination techniques. First, sample collection is absolutely critical for obtaining accurate results. It's essential to collect samples from representative locations at the right times to get a true picture of the microbial community. Always use clean, sterile containers and make sure to label each sample clearly with the date, time, location, and the name of the person collecting it. Proper storage is also vital. Typically, samples should be refrigerated at 4 degrees Celsius and analyzed within the specified holding times to prevent changes in the sample composition. Moving on to preparation methods, for activated sludge, we usually collect a mixed liquor sample and examine it either directly or after dilution. When working with trickling filters, we often collect biofilm directly from the media or examine sluice material found in the effluent. For rotating biological contactors, or RBCs, we scrape biofilm from the disc surface. To concentrate microorganisms, techniques like centrifugation, filtration, or settling are commonly used, which help improve the accuracy of our microscopic examination. Next, let's talk about staining techniques. The Gram stain is fundamental because it allows us to differentiate bacteria based on their cell wall structure. Gram-positive bacteria retain the crystal violet stain and appear purple, while Gram-negative bacteria take up the safranin counterstain and look pink. This distinction is crucial for identifying bacterial types. The Neisser stain, on the other hand, is used to identify bacteria that store polyphosphate. Cells that are positive for this stain will appear dark blue to purple, giving us insight into their storage capabilities. Now, on to enumeration methods. Counting chambers, such as the hemacytometer or Sedgwick-Crafter cell, are standard tools. We examine a known volume under the microscope and count organisms in specific fields to calculate concentration. For filamentous organisms, we measure the total filament length using a calibrated ocular micrometer. Sometimes, We assign a subjective score on a scale of 0 to 6, based on how abundant the filaments are relative to the flock particles. Finally, quality control is essential to ensure our results are reliable. We perform duplicate counts for precision, analyze split samples to verify accuracy, and include blank samples to detect any contamination. Maintaining detailed records of all procedures helps track consistency. Regular calibration of equipment and following standard methods are also key practices to uphold quality in our microscopic analyses. By mastering these techniques, we can obtain accurate, reliable data that supports effective wastewater treatment process, monitoring, and optimization. So, to wrap up, let's talk about the Microlife ecosystem in wastewater treatment. Essentially, this process is driven by a complex community of microorganisms, bacteria, protozoa, and metazoa, that work together in a biological system. Understanding these tiny life forms and how they interact is crucial for running a treatment facility effectively and maintaining control over the process. Now, these treatment processes are like human-designed ecosystems where every microorganism has a role. Bacteria are the workhorses, breaking down organic matter. Protozoa act as natural controllers, keeping bacterial populations in check. And metazoa contribute to polishing the water and helping stabilize the system. Each group has a specific function, and their interactions are what make the entire process work smoothly. One of the key tools we use to monitor this microscopic world is microscopic examination. It gives us immediate feedback on what's happening inside the system. By observing which organisms are present, absent, or active, we can gauge how well the treatment is progressing, whether the system is overloaded, or if there are emerging issues. Regular monitoring allows us to stay ahead of potential problems and optimize the process proactively. Operational control hinges on understanding and influencing these microbial communities. Factors like dissolved oxygen, mean cell residence time, nutrient levels, pH, temperature, and loading rates all impact microbial activity. When operators understand what these microorganisms need and how they respond, they can create the ideal conditions for treatment and prevent issues like bulking or foaming. And of course, this field is constantly evolving. Wastewater microbiology is dynamic, with ongoing research revealing new insights, whether through advanced microscopy, molecular techniques, or deeper process understanding. Staying current with these developments is essential for operators, engineers, and scientists alike, as it helps us improve and innovate our treatment processes. In essence, the microscopic world in wastewater treatment is a fascinating example of applied ecology. By harnessing natural processes and managing these microbial communities carefully, we not only protect public health and the environment, but also recover valuable resources. Success in our work depends on understanding and collaborating with these invisible but vital partners in treatment.